Hello everyone, my name is Arun. I am a PhD student at the University of Michigan, advised by Professor Rituparna Das. I'm excited to present GenomicsBench, our recent work in collaboration with Intel Labs that aims to study and characterize the performance of recent genomics applications. Genomics data is soon expected to hit the exabyte scale by the year 2025. The two major sequencing milestones were won by Illumina in the year 2008, which pioneered second generation sequencing, and the second one was by PacBio and Oxford Nanopore, which pioneered third generation sequencing. And ever since then, sequencing data has been doubled according to estimates by Illumina. In the contrary, computing capabilities, as governed by Moore's law, has been doubling only once every 18 months. Over the last decade and a half, the cost to sequence a single human genome has dropped by more than 1000x, from about $1 million in 2008 to less than a few $100s today. With the decrease in sequencing cost, there has also been a 1000x increase in sequencing machine throughput. Along with this, there has been a 1000x increase in the length of a sequencing fragment, also referred to as a read. However, the error rate per base has increased 10 to 100 fold. GPUs and custom hardware are also increasingly being used to accelerate sequence analysis, such as efforts from NVIDIA Parabricks, Microsoft and Stanford University, as well as Intel and Illumina. To fill this gap and to enable future architectural exploration specific to genomics, we propose GenomicsBench. GenomicsBench consists of 12 computationally intensive kernels which are drawn from well-maintained software tools. They are carefully chosen to account for the major steps in modern sequence analysis pipelines. They include both short read and long read analysis algorithms, and also a set of small and large input datasets are provided. Here's the outline for the rest of the talk. We have so far seen and motivated why we need a new benchmark suite in genomics. Next, we will see some modern sequence analysis pipelines. One of the most common modern sequence analysis pipeline is that of reference-guided assembly. In reference-guided assembly, we aim to reconstruct a sample's genome by aligning fragments of it to a reference genome such as the human reference genome. So the first step is sequencing, which in the case of Illumina sequencing machine is a set of fluorescent images of the DNA fragment, or in the case of nanopore, a set of current measurements as the DNA passes through the pore. In either case, these measurements, either fluorescent images or current, are now converted to a digital format consisting of characters A, C, G, and T, and these fragments are also called as reads. Once you obtain these fragments, they are now aligned in a computationally intensive step called read alignment, and after alignment, the differences between the aligned reads and the reference genome at a certain location are used to call variations. For short reads, Bustard is the common base calling tool for Illumina reads, followed by aligning WMM2 and GATK haplotype caller. Only 30 to 40% of the overall pipeline runtime. For long reads, Bonito is the open source base caller from Oxford Nanopore, followed by aligning reads with Minimap2 and calling variants using a neural network based tool called Claire. The second analysis pipeline is that of de novo assembly. In this case, the sample's genome is unknown. As a result, followed by sequencing and base calling as before, now we try to construct overlaps between these reads and try to reconstruct the genome from scratch. So we try to lay out reads that have significant overlaps to each other and then construct a consensus sequence, followed by an optional error correction or polishing step, which aims to correct small errors in the consensus assembly. With regard to software tools, Bonito is again used for base calling. Fly or Minimap2 can be used for overlap layout consensus, while error correction polishing is typically performed using a Racken or nanopolish pipeline. The third common analysis pipeline is that of metagenomics classification. In this pipeline, the users wish to analyze a soil sample or a gut or throat microbiome sample and try to determine the abundance of different microbes present in the sample. So similar to the previous two pipelines, after sequencing and base calling, we now align not only against one reference genome, but against a pan genome, which consists of the set of all bacterial and viral genomes as well as the human reference genome, and then try to find how many reads map to each genome and estimate. We first observed that dynamic programming is one of the most time-consuming steps in many genomics pipelines. As a result, we chose 5 of these kernels in GenomicsBench. These contribute 30 to 75% of the execution time in different software tools. These 5 kernels are shown here, and all of them have diverse compute requirements. The data dependencies in these dynamic programming or DP kernels can either be linear or two-dimensional. The inputs can either be sequences or graphs aligned to sequences, and the computation performed could be in integer domain or floating point domain. One of the simplest dynamic programming benchmarks in GenomicsBench is chaining. In chaining, given a set of reads and matching segments between them called anchors, we determine a collinear chain of anchors so that we can stitch together these nearby segments into larger overlapping regions. The algorithm tries to determine the best chaining predecessor for a given anchor by comparing against all previous predecessors and trying to determine the best one based on dynamic programming. This algorithm takes into account the number of matching bases between the anchors, the gap between the anchors, as well as the anchor length. The next dynamic programming kernel in GenomicsBench is the banded version of the popular Smith-Waterman kernel. In Smith-Waterman, given a set of query sequences and set of reference sequences, we try to find the best alignment between each query reference pair. Commonly, the query and reference sequences are assigned to different SIMD lanes, and dynamic programming is used to find the best alignment between each query reference pair. To compute the score in each cell, we require the score of its 3 immediate neighbors as shown on the right. Also, each cell in the H, E, and F matrices only involves integer computation. Next, we look at pairwise hidden Markov model computation in GATK's haplotype caller. The hidden Markov model consists of 3 hidden states, one for an insertion, one for a deletion, and one for a match between any 2 bases in the set of sequence pairs. The dynamic programming computation is parallelized across different SIMD lanes. The data dependencies are similar to the banded Smith-Waterman that we have seen earlier. The only difference is pairwise hidden Markov model aims to compute a score or a probability score for the set of all alignments between sequence 1 and sequence 2 in floating point, as a result of which this max is replaced by sum in this kernel. Let us take a look at the partial order alignment kernel from Recon, which is used to compute a consensus sequence from a set of read chunks. We are now looking at an intermediate step in the computation where we wish to align read sequence read 3 to a graph constructed already, graph 1 2. The data dependencies in this computation are slightly different from banded Smith-Waterman in that to compute a given cell, we not only need its immediate 3 neighbors, but also require all predecessors as defined by the graph topology. The DP computation is also parallelized across different columns by computing different cells in a vectorized fashion. The consensus sequence for the read chunk is shown here. We now describe some of the machine learning benchmarks that we have chosen as part of GenomicsBench. The machine learning kernels typically run on GPUs and have fairly regular compute patterns involving dense matrix multiplication in the floating point domain. So let us look at the NN base kernel, which is extracted from the base caller Bonito. This kernel takes as input the raw signal described as current measurements and passes it through a convolutional neural network, which then aims to determine the probability for a given base. We pass the result of the CNN through a CTC decoder that aims to determine the best path through the set of base calls so as to more accurately account for duplicates and determine the base call sequence. NN variant, which is part of the long read variant caller CLAIRE, aims to compute the probability of different variants given a set of aligned reads and then the reference sequence. To do this, it computes a set of input features which not only take into account the counts of different bases at the candidate variant site, but also a context consisting of 16 bases to the right and 16 bases to the left of the candidate location. These set of input features are passed on to a recurrent neural network which estimates the probability of different variants such as substitutions, insertions, deletions, and their respective lengths. Let us now take a look at one of the memory-intensive benchmarks in GenomicsBench. So given a reference sequence, the FM index of the reference sequence is defined as the set of cyclically sorted suffixes, as shown here, and the respective positions in the original reference sequence in a compressed representation. So for the human reference genome, the FM index of the reference takes about 10 gigabyte space. One of the attractive features of the FM index is that it can identify all the locations of a pattern read in the reference sequence in time which is proportional only to the length of the reference sequence. However, as shown here, this involves a series of irregular memory lookups, one per character, and this has very little temporal or spatial locality and resulting in high memory bandwidth requirements for FM index search. We now characterize the performance of the GenomicsBench benchmarks. Before diving into the microarchitectural performance characteristics, we first attempt to describe the available sources of parallelism in these benchmarks in terms of their high-level algorithm characteristics. We notice that several of these benchmarks operate on either reads or read batches or genome regions. And the work done per read batch or genome region in the case of dynamic programming benchmarks is typically an update to a given cell. However, since each read or genome region can be computed independent of each other, there is abundant data parallelism available in the order of billions for reads and in the order of millions for genome regions. However, we notice that there is significant variation in the amount of work done per task, say a read or a given genome region. We notice that across the different benchmarks, the ratio of maximum to average work done can vary by as much as 4.1 to 8.3x. We also see that when plotting the work done against its probability distribution, we obtain a normal distribution with significant variation. We now dive deep into the sources of imbalance in one of our kernels, the Bandit-Smith-Waterman kernel. We found that the SIMD utilization for this kernel was less than 50%. As you can see, the SIMD utilization by increasing the batch size in terms of the number of query sequences and reference sequence pairs only saturates at about 48 to 50%, even when using very large batches such as 1 million. Some of the reasons for the low SIMD utilization are early termination of the computation of the DB matrix in different SIMD lanes due to poor alignment between the different query and reference sequences. Furthermore, there is significant imbalance in the length of query and reference sequences, which can also contribute to different amount of computation that's performed in different SIMD lanes. We now look at the instruction diversity present in the different GenomicsBench benchmarks. This can help guide specialization efforts in hardware. We observe that three benchmarks, Bandit-Smith-Waterman, PAR-HMM, and partial order alignment use vectorized implementations and involve a significant number of vector integer instructions. Also, scalar integer computation is very common. Bottlenecks, as reported by Intel vTune's top-down analysis, we see that memory-bound benchmarks such as FM index and k-mer count, which involves looking up and updating the counts of different strings of length k in a hash table, involve significant amount of memory-related stalls. Compute-intensive benchmarks such as Smith-Waterman, PAR-HMM, and chaining spend more than 50% of their cycles retiring instructions, which is a good sign. When looking at the cache misses, we see that compute-intensive benchmarks have low last-level cache MPKI and spend less than 20% of the cycles waiting for input data. In contrast, the memory-intensive benchmarks, as expected, have moderate to high L3 MPKI. To summarize, the GenomicsBench benchmark suite consists of 12 representative kernels spanning the major steps in both short-read and long-read analysis pipelines. We have shown that while many of the GenomicsBench benchmarks have abundant data parallelism, they have significant irregularity, leading to imbalance in computation and making them hard to optimize for commodity hardware. This motivates the need for more vectorization-friendly algorithms as well as hardware that can deal with irregular data parallelism. GenomicsBench is open source and under active development.